Cane toads were introduced to Australia from Hawaii in June 1935. They were introduced to replace the use of pesticides like arsenic, pitch, and copper. The success of using moth Cactoblastis cactorum in controlling prickly pears in Australia also contributed to hopes for the cane toad. Since their release, toads have rapidly multiplied in population and now number over 200 million. They are known to spread diseases affecting local biodiversity. Unfortunately, the introduction of the toads has not only caused large environmental detriment, but there is no evidence that they have affected the cane beetles they were introduced to prey upon. Cane toads have negatively affected the biodiversity due to the fact they do not have a natural predator within Australia and multiplied rapidly. Cane toads were brought to Australia in 1935 to northeastern Queensland as an experiment to control a beetle that devastated sugarcane crops. This experiment was a failure because instead of controlling the beetle population, the initial 102 young toads quickly multiplied until there were hundreds of millions, stretching from coast to coast. Instead of going after the beetles, as growers have planned, the cane toads began going after everything else in sight: insects, birds' eggs, and even native frogs. And because the toads are poisonous, they began to kill would-be predators. The toll on native species has been immense. Cane toads soon became an invasive species in the area, even evolving to become better invaders by growing longer legs and becoming more aggressive, driving some species to extinction in local areas. The diet of cane toads is much like the proclaimed seafood diet. If a cane toad sees something, they will eat it. As long as it is small enough to fit inside their mouth, including a variety of invertebrates, reptiles, and birds, cane toads will consume anything in their path. This has caused the depletion of native fauna in Australia, but this depletion could be circumvented by eliminating the population. The complete eradication of cane toads in NSW is not feasible given their ability to thrive in a broad range of habitats, their capacity to reproduce in large numbers, and their current widespread distribution. Also, there are insufficient resources to control cane toads effectively in all areas where they coexist with native wildlife. There is no single solution to the cane toad problem. However, with concentrated efforts, the dilemma can be eradicated from Australia, though some scientists claim this is no longer possible. Cane toads are highly toxic. When the proposition to introduce cane toads to Australia was proposed, some scientists and naturalists advised against doing so. However, their warnings were ultimately ignored. A number of strategies have been implemented, one being of entrepreneurial direction. This plan exports the cane toads to the Chinese market for medicinal properties. Though seemingly a good idea, this is not massively effective in the eradication of the cane toad species. The most promising solution to the cane toad problem in Australia is further research in gene modification technology. Prior to their halt on it, scientists planned on creating a cane toad specific disease. However, they prohibited further research because of the disease possibly transmitting to other toad populations in other continents. Therefore, rather than using gene modification to create a disease for the cane toads, scientists such as Professor Peter Koopman and his team have devised a plan to eradicate the species working at the cellular level, instead creating a generation of cane toads which will only give birth to male offspring, diluting the population and eventually eliminating the species with the daughterless gene. The limitation of this strategy is that it is particularly costly and is offered as a long-term solution. Another strategy to aid in the eradication of the cane toad population in Australia is using parasitical lungworms specific to the toad and the utilization of an alarm pheromone which is given off by stressed tadpoles, causing interruption in the development in adolescent cane toads. This method is what scientists claim to be the most promising in solving the issue. The eradication of the cane toad population on Australia would allow for native species to once again thrive. Due to the depletion of native species that die eating cane toads, the poisoning of pets and humans, the depletion of native fauna preyed on by cane toads, and reduced prey populations for native insectivores. If the depletion of native species that die by eating cane toads increases, it may be possible that we continue to see the extinction of other species in Australia rather than the cane toad. Therefore, by ridding of the cane toad population, we may live to see native Australian species outlive the invasive cane toads. Furthermore, bufotenin, the deadly toxin comprising of the cane toad's toxicity, creates problems apart from the depletion of native species. It also proves to be a danger to pets and humans, who frequently are poisoned by the cane toads. Young children and pets are especially susceptible to the poison of the cane toads. If cane toads were eliminated from the Australian population, we would see safer environments for humans and pets alike. Moreover, solving the cane toad problem would lead to an increase in biodiversity by no longer disrupting multiple areas of many different food chains, allowing native plant and animal species to thrive. All of these effects on Australian ecology would disappear if we corrected the problem, resulting in a more biodiverse country and world.